In part 2 of chapter 6, I'm going to tell you about the powerhouse of the cell, by which I mean the chloroplasts. Also, the mitochondria, which, if you don't have chloroplasts, are also good. Chloroplasts and mitochondria share a lot of features in common. First and foremost, their function in cells. They are the organelles where energy is converted from other forms into cell happening, or energy transformation. The end of this unit will be all about energy transformations, 3 chapters worth. But we'll start out here with just the basics of mitochondria and chloroplasts and throw in some peroxisomes as well. One single mitochondrion, or powerhouse of the cell. Anyway, the mitochondrion is the star of chapter 9, so we'll describe the function in gory detail in that chapter. For now, let's get acquainted with the anatomy of a mitochondrion. The mitochondrion has 2 membranes, though it is not part of the endomembrane system. There is an outer membrane, which is smooth, and then there is an inner membrane, which is folded and convoluted in order to maximize the surface area. The folds of the inner membrane are called cristae, and the space inside the inner membrane is called the matrix. That's what you see here. Between the 2 membranes is a space called the intermembrane space. You should make sure that you enunciate that because there is an inner membrane and an intermembrane space. Also note that inside the matrix there is a loop of DNA and some ribosomes. Kind of a weird place to find them, but I'll explain that later in the lecture. Chloroplasts are found in plants and algae and use chlorophyll to collect light energy from the sun. We will see a lot of the chloroplast in chapter 10. The chloroplast in plants have 2 membranes like the nucleus, but in this case they surround a special area called the stroma. In the stroma is another level of membrane which is arranged as flattened discs called thylakoids. The thylakoids are in stacks called grana, or just one is a singular granum. The discs shape help to increase surface area, similar to what we saw with the cristae in the mitochondria. In the stroma, again, we find a loop of DNA and ribosomes. And now, why that is. The idea is called endosymbiont theory, and it goes something like this. Back at the very early dawn of the eukaryotes, a cell absorbed a bacterium, and instead of digesting it as food, the bacterium became engulfed, like a little cellular pet inside the eukaryote. Over time, the host and the pet became one. They merged into a single organism, the true eukaryotic cell with mitochondrion. Later, but still early on in the evolution of eukaryotes, other cells adopted a second bacterial cell, a photosynthetic one that evolved into the chloroplasts. Here's a drawing of what that might have looked like. An early eukaryotic cell with an endomembrane system engulfed a bacterium that became a mitochondrion. A second lineage of eukaryotes engulfed another cell, this one a photosynthetic bacterium. This second cytoplasmic pet was the ancestor of the modern chloroplast. The evidence for endosymbiont theory, like other theories, is significant and explains much of what we observe about chloroplasts and mitochondria. Chloroplasts and mitochondria each retain a small chromosome and their own ribosomes. We can connect the mitochondria and chloroplasts with their parental lineages within the bacteria. Chloroplasts are derived from photosynthetic cyanobacteria, and mitochondria from an order of alpha proteobacteria called the Rickettsiales. We also see that other eukaryotic cells can harbor living bacteria within their cytoplasm. For example, bacteria in the genus Wolbachia are found inside the cells of arthropods and nematodes very frequently. And one more piece of evidence, for now, We have seen eukaryotic cells that can accommodate other eukaryotic cells in a process called secondary endosymbiosis. One more organelle which isn't part of the endomembrane system is the peroxisome. The peroxisome sounds similar to a lysosome, at least when you look at their description of what they do, but it's not the same. Peroxisomes produce hydrogen peroxide, which is H2O2. Hydrogen peroxide is much more reactive than water. We use peroxide in that brown bottle on your shelf as a disinfectant for minor cuts. It bubbles as it kills, and then it is converted into water. Peroxisomes can be used to absorb reactive oxygen species, so-called free radicals. Another thing that peroxisomes are good at is breaking down fats. But if your eyes turn into dollar signs thinking they could be exploited for a simple weight loss solution, calm down. Weight loss is not such a simple thing. Seeds of plants are often rich in specialized peroxisomes called glyoxysomes that can break down the plant oils to release energy for the embryonic plant. Peroxisomes are recognizable because they contain a large protein crystal, which is geometrically incongruent with its surroundings, like you see here. Everything else is kind of round or flat, and the peroxisome protein crystal is a square. Another feature that makes eukaryotic cells more complex than prokaryotic cells is the presence of a cytoskeleton. We call it a cytoskeleton, though it's not made of little microbones, though that would be adorable. The cytoskeleton is made of different proteins throughout the cytoplasm that give the cell structure and connect activities going on in the cell. There are 3 types of cytoskeletal elements, which is not a large number for us to remember, fortunately. And they come in 3 sizes: small, medium, and large, each of which has different functions for me to tell you about. Can you guess what kind of microscopy produced this image? It's a fluorescence image with 3 different stains applied. The green stain is bound to the microtubules, which are the large cytoskeletal elements. The orangey-red stain is bound to the smallest ones, the microfilaments. And the blue is DAPI, which binds to DNA. The medium-sized intermediate filaments are not stained in this image. It's still a very nice image, though. The cytoskeleton serves to keep the cell's shape and as the transit system to guide all those vesicles and other structures to get to the correct places, as you can see in this drawing and SEM image. Again, as if it were so important I needed to mention it several times, there are 3 main components of the cytoskeletal system: the microfilaments, the intermediate filaments, and the microtubules. Microfilaments are the smallest in diameter, and they're also called actin filaments after the protein that comprises them. Actin may ring a bell, as I mentioned it as the example of a protein associated with movement back in Chapter 5, in conjunction with myosin. Actin forms a filament or thread, not a tube, so its forte is in resisting tension rather than compression. Microfilaments are most abundant around the cell cortex, or the outermost parts of the cell near the plasma membrane. They assist with cell movement in structures like pseudopodia, cytoplasmic streaming, and they also have a role in animal mitosis that we'll see in Unit 3. In this slide, we can see a few of the functions of the actin microfilaments. We saw in Chapter 5 how actin and myosin work together as the molecular basis for muscle contractions. Actin filaments also coordinate to allow for single-celled eukaryotes, like amoebas, to move by forming pseudopodia. And actin filaments also make cytoplasmic streaming possible, as we see in plant cells circulating their chloroplasts. Intermediate filaments are the medium-sized elements of the cytoskeleton, as if the name didn't give that away. They are also filamentous rather than tubular and made of diverse proteins, including different types of keratins. The intermediate filaments are stained orange in this micrograph here. The most important function that they have for our purposes is in forming the nuclear lamina. Remember when we saw that woven protein lining the nuclear envelope? Remember this slide from part 1? That is all intermediate filament. Microtubules are memorable because they are microscopic tubes. They are hollow like a paper towel tube and have a lumen, which you should remember means there is a space in the middle. The diameter is not so important except to remember that it's big relative to the other two. The protein subunits are globular proteins called alpha and beta tubulin. To make the tubes shorter or longer, tubulin units are added or removed. The function of the microtubules are to resist compression. Tubes are good for that. If you hold a cardboard cylinder between your hands like a paper towel tube, so your hands block off the lumen, then it resists your efforts to compress it. As we saw a few slides ago, the microtubules act like a monorail system for moving vesicles around when motor proteins are attached. They also drive eukaryotic flagella and cilia. I'll say more about microtubules in Unit 3 because they also feature prominently in cell division. But we're not done singing the praises of microtubules just yet. As structural, transport, and mobility members, microtubules require special handling. They require an MTOC, or Microtubule Organizing Center, to coordinate and regulate them. The centrosomes, made of a pair of centrioles, do this task in animal cells. The two centrioles are at right angles to each other, and each centriole is made of 9 units of 3 tubules, or 9 sets of triplets. As for motility, microtubules make eukaryotic flagella and cilia go. Remember, eukaryotic flagella and prokaryotic flagella are different in some very fundamental ways, while cilia and flagella in eukaryotes are very similar. Remember, the shaft of the bacterial flagellum was a solid protein rod that whipped around like a weed whacker. In eukaryotes, the flagellum is more like an animal's tail, with a plasma membrane sheathing a complex array of microtubules that are cross-linked by a motor protein called dynein. Inside the cell, at the root of the cilium or flagellum, is a basal body with the same 9 triplets that we saw in the centrioles. In the shaft of these structures, it switches to a 9+2 arrangement, so there are 9 pairs of microtubules and another pair in the middle. If we were to take these cross-linked doublets without a basal body and gave them ATP as an energy source, the dynein proteins would walk the doublets in opposite directions. In a working flagellum with a basal body, the cross-linking of microtubule doublets affects the wave-like motion that is what makes the structure capable of swimming. To wrap up our tour of the cell, I'll tell you about some special features on the outside of cells and how cells communicate with each other. In large multicellular organisms like you and I, the cells have to work together, and that takes some additional equipment. Let's start with a familiar one, cell walls. We've already seen that lots of different kinds of cells have cell walls, and that cell walls can be made of lots of different kinds of things. Peptidoglycan, pseudopeptidoglycan, chitin, cellulose are all cell wall components in different organisms. You could say that we, as animals, are the odd ones out, not having cell walls. All plants have cellulose in their cell walls, with some other molecules mixed in, but woody plants have an additional polymer in their cell walls. In these images, you can see that each cell produces its own cell wall, and they're all connected to each other by a structure called the middle lamella. In animals, the cells are connected together by an extracellular matrix, or ECM, comprised of different proteins like collagen, more hybrid molecules like proteoglycans, and fibronectin. In Chapter 7, we will learn more about integrins as well. In this illustration, you can see that outside the plasma membrane is a rich tapestry of different molecules that anchor the cells together. One type of molecule we find in the ECM are proteoglycans. They are different from glycoproteins, which are proteins with carbohydrates attached, in that proteoglycans have polysaccharides at their core with proteins attached. Multicellular organisms rely on the ability of the individual cells to communicate with each other and to exchange materials. There are particular types of junctions that make this possible. Plants have channels through the cell walls called plasmodesmata, one singular plasmodesma, multiple plasmodesmata. These channels allow plant cells to exchange materials with each other. Again, plants only. Animals have 3 types of cell-to-cell junctions for us to talk about. Tight junctions are long interconnections between adjacent cells that prevent leaking of the fluid from between cells. Desmosomes are like molecular rivets that allow strong epithelial sheets to form. And gap junctions are similar to the plasmodesmata in function. Both plasmodesmata and gap junctions allow materials such as small molecules, ions, and stuff like that to be moved from one cell to another. In summary, even though cells are made mainly of 4 different types of large biological molecules, and those molecules form into discrete organelles, a cell is more than just a series of parts, but a fundamental unit of life. Once again, our learning objectives, 1 through 7, 8 through 15, and that's the end.